Okay. Hello, everyone. My name is Rizal Hariadi, again from Arizona State University. First, I would like to thank Jing, Sonia, Ashok, and other organizers for this webinar. Congratulations. It has been many years, and you're still going strong, or we are still going strong. My lab is very excited to share our paper that was submitted on Tuesday to a journal. This is our new baby. It is on pulling biomolecules along multiple axes. And at the end of this presentation, we will show that, or we will share our surprising finding that when we pull biomolecules along multiple axes, the biomolecules of our interest, in this case, holiday junction, exhibit profoundly different dynamics. This work is being led by our current member and current graduate, current alumni, which is Bima here. This is him. And it is a wonderful collaboration with my colleague, Steve Perse at Arizona State University. The person who is leading this effort in his lab is Ayush Surab. Bima is right now interviewing for postdoc positions, and Ayush will be on the job market this cycle. And this work was supported by NIH New Innovator Award, and now is being supported by NSF Korea. And Bima was supported by American Heart Association. So I gave a talk about this work in Washington, D.C., so I'm just gonna reusing the introduction. I am a big fan of analogy. So imagine that we are in Washington, D.C., and we go to one of the memorials, in this case, Jefferson Memorials. But all we are allowed to do is just seeing the shadow cast on a wall, let's say. We will know that there are pillars, there is a statue, but we will miss a lot of important informations. We will miss the thoughtful expression of Thomas Jefferson. We will miss the orientation where he faced, which is he always faces White House. And we will miss the wisdom that is carved on the wall. Essentially, this is what many of us or scientists do. We try to get a full picture out of incomplete picture, out of the shadow. And this is where building new tools and building new analysis tools can be very helpful. And hopefully, and towards that goal, we want to make a contribution to the field by adding a new dimensions, pun intended, to the way we appreciate and understand nature. So let me be less philosophical and be more concrete. One example in biological physics is integrin here. These are integrins under different conformations. Integrins not only experience force along one axis, but it experience forces along multiple axes. So it interacts with extracellular matrix. It experience force from the outside environment of the cell. It also experience force along membranes. There is a membrane tension that may tune the function of integrin and also interact with proteins inside cell. These proteins are part of focal adhesion kinase. So nature is very complex. It is not confined on single axis. And this is different than many tools that we have benefited from. So our field, single molecule biophysics, has built and optimized many single molecule force spectroscopy, but all of this mainly work along one axis. What I'm showing right now on the left is atomic force microscope. It is basically a calibrated spring that mechanically connected to proteins of interest, and the whole thing is on an optical table, which is solid support. The one on the right is optical tweezer. It is basically beads in a tightly focused beam, and the beads has the lowest energy in the center of the beam waist. So any deviation from the beam waist would generate restoring force. So again, the take-home message of this slide is that all of these tools are powerful, high sensitive, but they are limited to single axis. And we know that cells and biology and nature doesn't just operate along one axis. I'm running my lab. I'm part of the Department of Physics at ASU, Arizona State University, but I'm also running a lab in a Biodesign Institute. So I encourage my students to come up with design principles of, let's say, biomolecular systems or equipment. And this is what we come up with when we survey a single molecule for spectroscopy. There are at least 3 design principles. One is calibrated spring. So AFM, the cantilever is the calibrated spring. The tightly focused beam of a laser with certain power is the calibrated spring for optical tweezer. Binders that connect the calibrated spring to the molecules and solid support. The nice thing with extracting design principles is that we can build a different equipment with out of completely different materials on a different scale. Right. So this is what Bima and other lab members in the lab come up with. It is a molecular tools that captures or that has all those 3 design principles. And our goal is to use this molecule as a low-cost, high-throughput device to stretch biomolecules along multiple directions and combine it with traditional assays such as can be gels, single molecule confocal FRET, or turf-based FRET, or even cryo-electron microscopy. Okay, so Bima came up with a nice abbreviation, which is multi-axial entropic spring tweezer along a rigid origami. Entropic spring is the calibrated spring. Rigid origami is the rigid scaffold or platform. Multi-axial is the number of axes where we can potentially pull biomolecules. And then we have to insert tweezer because based on when we survey tools, it seems that biologists love the word tweezer, and we want biologists to use our device. For completeness and complete transparency, we are not the first one who came up with this idea. Henrik Dietz at TMU Munich and Carlos Castro at Ohio State and their colleagues have started tinkering with using DNA origami, which I will explain on the next slide, as a device that can apply tension to biomolecules. So this is the device on the right panel, on the left panel. So the dimension is less than 100 nanometer. The second approach was published by Tim Liddell with his, I think, graduate student at the time, Philip Tinnefeld. They use entropic springs, and they published their work in Science in 2016, I believe. So we want to use DNA origami because it is the technique that we have in the lab to build a nanometer scale object with less than 5 nanometer precision. So DNA origami basically a facile, an easy way and a reproducible way to build nanostructures. What we do is we take long DNA strands. Typically, we steal that from a virus, a bacteriophage, and we know the sequence. And then from that, we can design short staple strands. And we order the staple strands. It will arrive in less than a week. We mix everything, and then we put salt. We heat it up and slowly cool it down. Close our eyes, and lo and behold, we get a beautiful structure like this. I didn't come up with this. The person who invented DNA origami is Paul Rothman. He was, or he is, still at Caltech. he was a postdoc in the lab where I did my PhD, or in the lab of Eric Winfrey. So for this work, the relevance of DNA origami is that we can use it to build molecular structures of defined shape. So we want a shape that is rigid and symmetric. So that's one. Second is that every short strand is unique. So we can potentially, or we can place entropic springs at any configuration of a defined number and defined spacing. Okay, so we want to control the strength of the entropic spring and also the directions of the forces. Okay. It is relatively easy to perform, and the structure is stable. This is an honest representation of Maestro, our device. This is a negative stain transmission electron microscopy image. And basically, it shows that the structure forms fairly well. It has the right dimension. So this is the one on the right is the cartoon. This is what we image, what be my image. And then it's not perfectly rigid, but it's fairly rigid. So that's good to know that our structure is not completely rigid. It is also not perfect. We have, let's say, linear structure here. Seems like almost a dimer here. But DNA origami was invented close to 20 years ago. So we know how to enrich the structures that we want and remove the structures or the side product of our assembly. So having established the size of the size and shape of origami, we want to assay the positions of, or the docking positions of the springs. So here BIMA designed 8 positions, and we use super resolution microscopy that use DNA. The name of the technique is DNA paint. It was invented in the lab of Fritz Simmel by his former graduate student, Ralph Jungmann, and further developed at Wyss Institute at Harvard in the lab of Peng Yin. So basically, this slide shows that we can control the position of the dockings. So let's say if we want to apply force along one axis, then we can connect spring along this side and this side. Or let's say if we want to do 2 axes, then we can place another spring along left side and right side. And these biomolecules can be in the middle. And we will show you data from that experiment. Okay. So having established that we can form the structures and we can tether the calibrated spring, now we need to decide what is the spring that we need to use. The natural choice for the spring is DNA, or single-stranded DNA, not only because the structure is DNA, that thing is very practical, very easy to do, but the hardest thing is that finding structure that is, or spring that is has been rigorously analyzed or assay. So the elasticity of single-stranded DNA has been measured by Carlos Bustamante and his co-workers. This is from their paper in 1992 in Science. So the horizontal axis is the separation between 2 beads. So they put DNA, long DNA between 2 beads, and they move one of the beads away from each other. So it turns out that single-stranded DNA is very nonlinear spring at low force from 40 femtoseconds to 15 piconewton, basically moving the end-to-end of long single-stranded DNA, doesn't deform DNA, but just orient. And what happened actually when they move the end-to-end distance is it changed the number of conformation that the extended spring can sample. So in a way, there is loss of entropy. And this is the reason why we pull rubber bands. It doesn't have to be DNA, like any linear or perhaps nonlinear polymers. When we separate, when we stretch it, there is a restoring force. This is because the system or nature wants to maximize entropy. Okay. So DNA, single-stranded DNA has been characterized. We know that it's an entropic spring, and we know that connecting DNA to DNA is very straightforward. So what BIMA is building is a structure that keep the end-to-end distance. So in Maestro, the end-to-end distance is 45 nanometer. And as long as we know the number of nucleotides of the single-stranded DNA entropic spring, then we can infer the tension along the spring. So this is a plot for 45 nanometer end-to-end distance. And BIMA used this formula here, presumably derived by Eric Sigia. Eric Sigia is another senior author of this paper. So it has number of nucleotides, it has temperature, and also it has end-to-end distance. Based on the length or the contour length of the entropic spring, we can infer the tension, which is the vertical axis. Okay. All right. So what we want to do is one step further. We don't want to just infer from optical tweezer. We want to also measure the effect. Okay. So to do that, we rely on kinetics experiment. If you're familiar with DNA paint, this is similar to DNA paint. But if you're not familiar with DNA paint, don't worry. So the kinetic experiment measures the association and dissociation rate of two complementary single-stranded DNA. And one of them is stretched along a Maestro. And the other one is freely diffusing in solution. And we label the freely diffusing single-stranded DNA with Psi3 with a fluorophore. So when Psi3 binds, we get high fluorescence. When the strand Psi3 labeled strand dissociate, we get no signal. So this fluorescence trace is basically dark, bright, dark, bright, dark, bright. From this, we can get the dwell time or the bright time and the dark time. And from the distribution, we can measure the K on and K off of the interactions between the imager strand in blue and the stretch docking strand in white. So you notice that there are two panels here, top row and bottom row. The bottom row is stretched at higher tension because the contour length is shorter. The contour length of the panel, the bottom panel, is less than the top panel. And that manifests, or the result is that the bright dwell time or the bright time is significantly shorter than the top fluorescence trace. So this built confidence to us that our entropic springs actually works. It does something, and we can tune the tension. So, and then we use a model that was derived by Harold Kim. He is a faculty at Georgia Tech, Department of Physics. He measured and built a model of association rate or K on and K off as a function of tension. In the interest of time, I will only talk about the dissociation rate. Basically, as expected, as we apply tension, we destabilize the bond, and then that will increase the dissociation rate. And based on the model, it should decrease exponentially. Okay, so our data fits Harold Kim's model, and then from the curve fit, and the fitted parameters also matches other experiments from other labs. Okay, so now we're ready to attach biomolecules. For the biomolecules, we use what we consider as the hydrogen atom for single molecule biophysics, which is the molecule that TJ Ha and TJ Ha's lab and other labs work 2 decades ago, which is holiday junction. It is an intermediate of homologous recombination. And we hypothesized that during that process, it experienced multi-axial tension. It is basically 2 double-stranded DNA that are connected by a junction. And the reason why it forms 2 cylinders because of this stacking interaction here. The stacking interaction is not is stable, but it's on the order of magnitude as thermal energy, so it can break. So when it breaks, the conformation on the left switch to the conformation in the middle. And then once holiday junction reach the conformation in the middle, the red can either stack with gold and return to the original state. We call this isomer 1, or can stack to green domain and becomes isomer 2, or the conformation on the right. Okay. And the way single molecule biophysicists study holiday junction is by conjugating a donor fluorophore and an acceptor fluorophore, so Psi3 and Psi5. The conformation on the left, which is isomer 1 here, has a higher frets, and the conformation on the right has a lower fret. And for this experiment, we will show you our data using TURF single molecule fret. But in the lab, we have been also doing this experiment using a confocal setup. So the experiments here, in these experiments, we immobilize Maestro with holiday junction and image it using TURF microscope. Just for completeness, basically, this is the data that we see. We follow DNA trace in a donor channel and acceptor channel. So when the donor signal is low, the acceptor channel is high. And also when the donor channel is high, the acceptor channel is low. And from this, we calculated the fret efficiency. Okay, so now I'm going to present the surprising results that we saw in the lab. So first, let's establish the baseline, which is F is equal to 0. So as a negative control, we monitor the fret trace of holiday junctions under 0 tension. There are many ways to do this control. We can just study holiday junction by itself, but we chose a different control. We chose to tether holiday junction, but with only one spring. So instead of a full pair, there's only one pair. So the holiday junction diffuse near, or basically tethered near Maestro, and we monitor the fret efficiency of holiday junction. Okay. So I'm going to show you data. If you are familiar with single molecule biophysics, single molecule measurement of holiday junction, perhaps you will feel a bit bored by this, but this is actually something that I didn't appreciate until we ran this experiment. So first, it turns out that when we run experiments of holiday junction with the same sequence, there is heterogeneity in the experiments. What I mean by that is that there are different kinetic classes of holiday junction dynamics. The dominant one for the one that we chose is class 2, but some molecules only show mostly isomer 1, iso 1, and some other molecules mostly only stay in isomer 2. Okay. This is actually, we're not the first one who saw this. Tim Liddell's paper, for example, has a sentence about this. But the first paper that fully characterized this dynamics is a paper by Dave Thromalai. He's at UT Austin. He published that paper when he was still at University of Maryland, Baltimore County. So basically, one sequence can exhibit different personalities or different dynamics. But for the one that we study, the dominant one is kinetic class 2. So this is, maybe it's easier to present as in the context of a network. So based on the Feromali's paper, holiday junction can exhibit different classes. And each class has at least 2 nodes. There is intermediate, but we will skip intermediate for now. And each class has 2 states, isomer 1 and isomer 2. But they are not connected, meaning once a holiday junction is in one kinetic class, it cannot jump. It cannot convert to other class. Okay. So this is the network representation of the dynamics that I show on this slide. So maybe another thing that I need to point out is that this plot here is the heat map based on Bayesian inference. This is based on our the result of our collaboration with Steve Rosset. So what we are plotting here is the escape rate from isomer 2 and isomer 1. Okay. And from this, we can compare the kinetics, and using k-means clustering, we can cluster different kinetic classes. So now we're ready to apply tension along holiday junction. Historically, we are not the first one who apply this tension. Tim Liddell did it along one axis using DNA origami, tension-inducing DNA origami for spectroscopy in 2016. There is another paper on this topic. The first one that I'm aware of was done by TJ Ha in 2007. It was a tour de force of experiments. It combines single molecule confocal and optical tweezer, single molecule confocal to measure fret efficiency, and optical tweezer to apply tension to a holiday junction here. So the question is that, can we simplify the results using high throughput low cost DNA origami? And next is, can we apply more than one tension along a single holiday junction? Okay. All right, so this is our setup. As you can, I hope you remember that we have 8 different docking sites. For the first data that I'm showing you, we apply tension along the vertical axis, along y-axis. So if this is holiday junction along y-axis, it can go to an intermediate state. This state is short-lived, and then it can go to low fret state. And we hypothesize that the low fret state is more stable because it's parallel with the direction of the force. Okay. And this is the data that we get for experiment with 0 piconewton, 2 piconewton, and 3 piconewton. The higher the tension that we apply, the more low state, the longer the holiday junction to stay in low state isomer or iso 2. Okay, so that's one. It also, when we measure the kinetics, we see changes as a function of tension. So the escape rate of ISO 1, which is high fret, increases. So that leads to decreased population of high fret, and the escape rate of the low fret decreases. That leads to an increase of fraction in low fret. But one thing that is important to note is that we only see 2 states. So our measurement is not sensitive enough to detect the intermediates. So there are only 2 states. So that's one. Second is that, similar to zero force experiments, there is no interconversion between class 1, class 2, class 3, and class 4. So it stays on one class. Oh, one thing that I forgot to mention is that I'm not showing all panels of the paper that we submitted earlier this week. there are more data. If you are interested in the bioarchive paper, if you are interested, I invite you to check it out. Okay, so this is the surprising result that I've been hinting in the last like 30 minutes. I apologize that we run longer than 30 minutes. So in this case, we apply tension along two axes, vertical axis and horizontal axis. So this is a cartoon of Holiday Junction. So the Holiday Junction is in the middle. The spring is represented as a thick blue arrows. Okay. So what do we see? So we see dramatically different results. So I'm just going to show you the comparison, and then we're going to return to the previous slide. So the one on the left is the data that I showed 2 slides ago, which is single axis, force along single axis. We don't see change in the dynamics, right? It always has the same let's say average fraction of isomer 1 and isomer 2, high Fred and low Fred. But when we apply tension along two axes, the molecules exhibit profoundly different dynamics. So let's say initially it exhibits class 1, which is equal proportion of high Fred and low Fred. And then it switched to a different class. And the time scale here is longer than 10 seconds, which is very long for biomolecules. And then it switched to a different slide again, kinetic class again. So this is a single Holiday Junction, and we have, I think for this experiment, we have 141 trajectories, and many of them show different kinetic classes. Okay, that we didn't see for experiment in single axis. Okay. So there is a term for this. So let me return to this. So here, if we take the mean of a measurement, let's say a fraction of, or the mean of Fred efficiency, then the mean of single trajectory is different than the mean of ensemble. But so there is a term for that in statistical mechanics, which is non-ergodic. So we think what happened with applying multi-axial force is that multi-axial tension restores ergodicity, or to be safe, to be proper, we call it restores quasi-ergodicity. The reason we don't go full ergodicity because our measurement is only 50 seconds, and we don't see trajectories that sample all kinetic classes, 1, 2, 3, 4, and 5. There are many other surprising results. For example, we can start seeing the new states with tension. And another result is that we see different kinetics. Okay. So the change of kinetic classes actually have been observed before by Dave Romalai, but he didn't do it without chemical perturbation. So what he did in his paper in 2012 is that he ran experiments at very high magnesium concentration, and then he stripped magnesium concentration using chelating agent, which is EDTA, and then there is no dynamics. And then he add high concentration of magnesium again, and he claimed that the Holiday Junction exhibit different dynamics. And the way he described dynamics is, one, as a function of a fraction of high Fred and low Fred, and also as the magnitude of Fred efficiency of high Fred and low Fred. And you can see that they are different here between this side and this side. So what we do is different. There is no chemical pulse. There is no chemical chemical perturbation. everything sits in a sealed cover slip. We just apply tension along two different axes. And what we think happened is that initially, without tension or with only single axis force, we see these different kinetic classes. But by applying multi-axial force, it smoothened the energy landscape, and it allows kinetic class 1, let's say, to jump into different kinetic classes. So the networks switch from an isolated classes to an interconnected network, like the one is shown on the right panel. I think in the interest of time, I'm going to show you one more result, and I'm going to skip a lot of details. Right now, what we are focusing right now is using Maestro to study protein functions as a function of tension. The first one that we try is T7 endonuclease 1. This protein breaks holiday junction into two double-stranded DNA. The name of this class of protein is junction resolving enzyme, or resolvase. And it has been studied by many labs, including TJH. But to our knowledge, all of the published work were done in the absence of tension. And we think during homologous recombination, this holiday junction experience multi-axial tensions. So this is a figure from the manuscript that we submitted on Tuesday. In the interest of time, I'm gonna skip a lot of details, but basically, the take-home message of this slide is that with tension, so we vary the tension from 0 to 6, and then we characterize the change in the junction as a function of whether the enzyme was there or not, and whether the tension was there or not. And we found that based on tension, the resolution of the junction or the cutting of a junction can result in the product on the left, where the red strand is not cut and the green strand is cut, or the product on the right, which is the product with intact green strand and cut red strand. So the main result is on the bottom right panel. Basically, the fraction of the green strand that get cuts is as a function of tension. And similarly, the fraction of red strand that get cut is also a function of tension, but they exhibit the opposite behavior that we think should be expected. So with tension, the red cut, the product that cut red strand wins at higher tension. I think this is the summary slide. So basically, we show our molecular tools that can induce tension along multiple axes. And we built this tool using design principles of large-scale force spectroscopy, which are rigid platform, calibrated springs, and binders, or mechanical connection between biomolecules and the molecular tools. In terms of the physics that we saw that holiday junction dynamics is non-ergodic, which reproduce results from many labs. And the probability of high Fred state and low Fred state depends on the direction of the force and also the magnitude of the force. And then one thing that we are very excited is that multi-axial tension restores quasi-ergodicity. And this is my acknowledgement slide. I've touched the names in the beginning of the talk. And yeah, thank you for listening. Look forward to your questions.
All right, let's thank Rizal for the very
Thank you.
interesting, very nice presentation. So if you have a question, please either just unmute yourself and ask, or you can put your question in the chat if you don't have, audio. Yay. So right now in the chat, I think we have an early comment from Boris. Plato had a similar insight. I think Boris was referring to one of the philosophical slides uh, related to the Jefferson
Okay.
Memorial.
So I had a
Yeah,
beautiful
typically I match
talk.
that with okay, go ahead.
Yeah, beautiful talk, and yeah, such an amazing
Go ahead.
technique. I had a question about your three design principles. Could you just repeat them again? Somehow there's only one that's stuck in my head.
Okay. So this is my design principles. This is
Yeah,
not
exactly.
a
Yeah,
published work. One
yeah.
is calibrated springs. This is either FM cantilevers or magnetic field or the focus beam or in optical tweezers. So that's one. Second is rigid platform. This is why we put a lot of masses on
Hmm.
the side of Maestro, or this is why we put magnetic tweezer on an optical table. And the last one is just mechanical connection. I call that binders because in our current work, we have binders that grab integrins or other molecules. So those are three. One can
Right.
say that it needs to have feedback, and feedback maybe doesn't have to be electronics. It can be mechanical. That could be another one. But for this talk, I only mentioned three.
Thank you.
Okay, thank you. Nancy
Yeah.
has, okay, hi.
Hi, thanks. That was really interesting. And I love the idea
Thank
of
you.
pulling from multiple directions at once and being able to track multiple single molecules at once as well. I was curious about the mechanism by which you think that the class switching is possible when you apply tension multiple places. Like, do you think that that enhances like transient dehybridization or other things, or is it somehow, I don't know, altering how easy, like, because I could imagine the surface might play a role in
Yeah,
locking in conformation. So wondering what you're thinking about there.
there are many hypotheses or possible explanation in the lab. The easy, I wouldn't say easy, but the experimentalist possible explanation is it could be that things stick to things. So that's one, right? But we did our best with our surface specification. We can improve it. We can reorient the Maestro to make it less likely to bind to surface. So that's one. But I'm gonna switch to a different explanation, which could be that by applying tension, we can decrease the dwell time of trapped cations on the junction. So people have seen that in molecular dynamic simulation that for potassium, this is because doing molecular dynamic simulation with divalent cation is more difficult than potassium. Once potassium sticks, then it always sticks during the course of molecular dynamic simulation, which is not long, not 20 seconds, but microsecond, 10 microsecond, perhaps. So we think magnesium in the junction is trapped much longer, and by applying tension, that magnesium can dissociate faster. And people found that depending on the coordination of magnesium in the junction, Holiday junction has different dynamics. So that's one explanation. That requires a long dwell time of magnesium, which we can only extrapolate from molecular dynamic simulation. I don't know how to measure it. So that's one.
That's a really interesting idea. Sorry, go ahead.
Sorry. A naughty explanation is by my PhD advisor is that this spring can entangle. And when it entangle, it has different direction and magnitude of forces. But I think thermal energy is so violent, I don't think that thing can stay for like 30 seconds. So it's possible, and we're running coarse grain simulation to evaluate that possibility and the dwell time of entangled springs. But I think that thing is not likely, but we need to finish the simulation first.
In
And
terms
then
of
another
the,
thing, oh, go ahead, please.
no, I was just curious, and if anyone else has questions, interrupt us. But I know people can do like luminescence resonance energy transfer using heavy metal ions, and I don't know if those could bind to your holiday junction similar to magnesium, and whether you could use a similar sort of fluorescence or FRET type approach to actually look at dwell times of those ions, but might be interesting.
Okay. Yeah, we haven't really explored that. But there will be, I would love to measure dwell time of magnesium in holiday junction. But I'll keep that thing in mind. Thank you for the suggestion.
Yeah. Really nice work.
Thank you.